It is my great pleasure to introduce our first speaker, Dr. Yadira Soto-Feliciano. Yadira did her undergraduate degree at the University of Puerto Rico and her PhD with Dr. Michael Hammond at MIT, where she used functional genomics to understand human blood cancers. She stuck with this cancer theme, and Yadira is currently a postdoc with Dr. David Allis studying epigenetic regulations in cancers. Yadira has received many, many awards, and I'm not going to go through that entire list, but her most recent award was the Mosaic K99 Postdoctoral Career Transition Award. And even though Yadira is our trainee speaker today, she will be returning to Boston to start her own lab at MIT Biology and the Koch Institute in the spring of 2022. She is hiring postdocs and research technicians, and Yadira is absolutely an amazing person to talk to. So if you enjoy her work today, please get in touch with Yadira or stay for the coffee chat afterwards to talk to her about more of her work, her lab, etc. All right, with that, take it away, Yadira.
Thank you so much for the very nice introduction. And yes, I'm hiring for my own independent group. So if you're interested, please get in touch with me. Let me just close this here. So hello, everyone. First of all, I really want to thank the entire Fragile Nucleosome team for inviting me here today. This seminar series has been such an amazing platform, keeping us connected through our common interest in all things chromatin, even during these very hard times. So thank you so much. So now I will take this opportunity to share with you some of the work I have done as a postdoc in the Allis lab, trying to dissect the mechanistic roles played by chromatin adaptor proteins in regulating gene expression, and also how cancer cells can then co-opt these molecular processes to then sustain their survival. And as a cancer and chromatin biologist, I am very fascinated by the central question of what are the major fundamental differences, but also the relationships between genetic and epigenetic mechanisms, and how the dysregulation of these processes are then ultimately linked to human diseases such as cancer. And to start getting at this, I have turned my attention to understand chromatin and how it is regulated at the most basic level. And chromatin, which doesn't really need much introduction to this audience, is the physiological form of our genomes, and it is in essence composed of 4 core histone proteins and DNA that are wrapped tightly into nucleosome particles, which we will hear a lot about in the next presentation. And this structure is able to then facilitate but also restrict the access of the transcriptional machinery to specific genes, really serving as another mean of gene regulation that now lies outside of our genomic DNA. And by the efforts of many, many groups, including many presenters at this seminar series, we have learned that chromatin states are regulated by a variety of histone and also DNA modifying enzymes, chromatin remodelers, and other structural proteins, including chromatin adapters or scaffolding proteins. And this last group here, highlighted in red, includes the chromatin regulators that have been the main focus of my current, but also will be the focus of my future work. And I will come back to this in a few slides. So our current view in the field is that the concerted regulation of chromatin states is able to then serve as a signal integrator or a transducer of both external and internal stimuli, for example, changes in cellular metabolism or developmental signals, to then orchestrate what are known as chromatin-dependent functions, which include, for example, cell cycle regulation, genomic stability, and gene regulation. And in the context of cancer, we now appreciate that this disease or diseases actually arise in part due to profound dysregulation of gene expression programs that are both the consequences of genetic and epigenetic alterations. And more recently, we have learned through tumor sequencing efforts that chromatin and epigenetic factors are actually often hijacked and are frequently altered in cancer. And these observations really support a more causal role for chromatin and epigenetic dysregulation as another important driver of the cancer state. And as I said earlier, we as a chromatin biology community have made a huge progress determining both the composition and function of chromatin modifying and remodeling complexes. However, something important to mention about these studies is that they have mainly focused on the catalytic domains of these complexes, and they have uncovered actually a great deal of diversity, redundancy, and more recently, catalytic independent functions for these enzymes. So therefore, in order to get a more holistic understanding of the molecular functions and the recruitment mechanisms of these multi-protein complexes, I have focused on studying and understanding what are known as chromatin adaptor or scaffolding proteins. What are these and what do they do? Well, so these are the factors that are responsible for tethering, modifying, and remodeling complexes to chromatin to then regulate transcription and ultimately impacting gene expression. Interesting, these proteins are often pleiotropic in nature, and they can oftentimes exhibit context-specific functions, both in the normal and disease states. And for example, the same factor as highlighted here in these diagrams is able to assemble different complexes within the same cell, actually, and then regulate or impact gene expression in completely opposite directions. So then to understand how these chromatin regulators operate, we started by asking three important questions. So the first one is, where and how are these factors genomically distributed and targeted? Second, how can they recruit a particular set of effectors for specific functions? And lastly, how can they ultimately impact and regulate gene expression? So my postdoc work here in the Allis lab has been focused on understanding and studying the chromatin adaptor protein known as Menin. And this is an evolutionary conserved and predominantly nuclear factor that is able to associate with chromatin to then recruit or adapt, hence the name of chromatin adapter, interacting factors. And structurally, it is fairly simple. So it only has two nuclear localization signals and an intrinsically disordered proline-rich region that are proximal to the C-terminal portion of the protein. And very interestingly, Menin doesn't share any sequence homology with any other known proteins in the eukaryotic cells, suggesting that it has very specialized functions. And in addition, Menin has been described to actually mediate many protein-protein interactions with several key factors that are known to regulate many important cellular and molecular processes, including gene expression, where it actually has been shown to regulate both activation and repression of transcription. Having said this, we still don't fully understand how Menin is able to choose among such a diverse group of interactive proteins. And beyond these biochemical interactions, we still don't know how Menin functionally interacts with these in the cells. And in the context of cancer, we have learned in recent years that actually MEN1, which is the gene that encodes for Menin, is recurrently altered or mutated in human tumors. In fact, there's been over 1,000 different genetic alterations in this gene identified to this day. And something interesting is that the distribution of mutations is actually widely heterogeneous, as you can see here in this diagram. They're spread diffusely across the entire gene, and with the exception of a very obvious hotspot in this arginine 521 proximal to the C-terminus, which is actually a site for recurrent friendship mutations. And another aspect of Menin biology in cancer is that it can have context-specific functions or paradoxical functions. So in fact, Menin acts as a tumor suppressor in neuroendocrine tissues. And on the other hand, it has been shown to act as an oncogenic cofactor in acute leukemias, particularly those that are driven by translocations of the MLO1 gene. And this gene, the MLO1 gene, actually encodes for a histone methyltransferase of the same name. And this enzyme, along with the chromatin adaptor Menin, are actually part of the same complex, the trithorax-related MLO1 complex, which is known to catalyze both the di- or trimethylation of lysine 4 and histone H3, which is canonically associated with coactivation of transcription. So MLO1 translocations actually fuse the amino terminus of MLO1 to one of more than 80 different fusion partners, resulting in a chimeric protein that do no longer have the canonical enzymatic activity. And something interesting about these fusion proteins is similar to wild-type MLL, they actually rely on the obligate direct interaction with Menin to then elicit their oncogenic function. And because of this, there's been a lot of effort to develop small molecule inhibitors of this protein-protein interaction as a potential therapeutic strategy for leukemias that are driven by these chimeric proteins. So when we started this project, the proposed mechanism of action of these Menin MLO inhibitors in MLO fusion-driven leukemias actually was via the suppression of the leukemic gene expression programs that are known to be regulated by these chimeric proteins, which then results in terminal differentiation in cell death. So that will be the phenotype of these small molecules. So this led us to then hypothesize that targeted genetic perturbations will perhaps able to then revert these phenotypes. So then to test this, we employed a focused CRISPR-Cas9 screening strategy combined with a Menin MLO inhibitor to then identify novel Menin regulatory pathways and novel biochemical and genetic interactions that perhaps are associated with these chromatin adapter. And then in close collaboration with Scott Lowe's group at Memorial Sloan Kettering, we constructed a novel antiviral sgRNA library that targets roughly 600 chromatin regulators. We then screened this library in a leukemia cell line that was derived from an MLL-AF9 fusion-driven leukemia mouse model, and then cultured these cells in media containing the inhibitor. So we actually were interested in mechanisms that will allow these leukemia cells to bypass the anti-leukemic effects of these small molecule inhibitors. So we then focused on genes whose loss of function actually allow the cells to survive. So in this case, they will be enriched in the context of the inhibitor. So here I'm showing you the results from the screen from cells derived from the vehicle treated or the menin amyloid inhibitor. And what we found is that actually among the most significantly enriched guide RNAs were actually those targeting the histone demethylase UTX and the histone monomethyltransferases MLO3 and MLO4. And as many of you might be able to recognize, these three chromatin modifying enzymes are actually part of the same multi-protein complex known as the MLO3/4-UTX complex. And to us, this result was actually kind of unexpected because what we're doing in this system is actually we're perturbing the protein-protein interaction between MLO1 and menin in this complex, which is known to catalyze the ditrimethylation of H3K4 at promoter regions, while the MLO3/4 complex is actually known to serve as the major monomethyltransferase for the same lysine residue, but at enhancers. And I want to stress that these complexes are actually considered functionally and genomically distinct complexes. And what these findings are suggesting is that the core enzymatic subunits of the MLO3/4-UTX complex are actually able to modulate the therapeutic responses of leukemia cells to menin amyloid inhibition, really pointing to a previously unknown functional crosstalk between these two. And then given the interest overlap in chromatin and epigenetic regulation, we then asked the question of what are the molecular consequences of this novel epistatic relationship between these two complexes and how they influence the chromatin landscape. So then we perform a series of chip sequencing experiments to determine the genomic distribution of menin and UTX. And here I'm showing you the chip sequencing results for menin in the two conditions from cells treated with the vehicle or the inhibitor. And what we find is that menin is actually primarily enriched and also in accordance with previous studies at promoter regions, and that this enrichment is actually sensitive to the action of these menin amyloid inhibitors. And conversely, while we only detect a very minor fraction of UTX bound to chromatin genome-wide, the genome-wide enrichment of UTX actually dramatically increases in the context of the inhibitor. In fact, we actually observed a more than a four-fold increase in signal for UTX at promoters. And this result was kind of puzzling initially because, as I mentioned before, the canonical view in the field is that these two factors are actually the exclusive subunits or the adaptor proteins for these two complexes that normally reside in or regulate very distinct cis-actin elements. And what our data is suggesting is that disruption of the menin MLO complex is now inducing targeting of UTX and perhaps other subunits within the MLO3 complex to non-canonical sites. So then to better understand this drastic increase in UTX binding to promoters that is elicited by the menin inhibitor, we then took a close look at these promoter regions. So here I'm showing you again the chip sequencing data for UTX ordered by chip signal of UTX in the promoters that are acquiring this factor in the context of the inhibitor. So then we looked at menin chip signal at these very same promoters. And what we observed is that not only does menin depletion actually leads to recruitment of UTX to promoters, the loci that accumulate UTX actually greatly overlap with those where menin is lost, and suggesting that a previously unknown function of menin is, in fact, to restrict the chromatin occupancy of UTX at promoter regions. So we also examined the chromatin binding profile of the cognate methyltransferases for these two factors and their enzymatic products, and we also observed the same changes, and I won't have time to show that data today. And what our data is really suggesting is that disruption of the MLO1 menin complex now induces retargeting or redistribution of the enzymatic subunits of the MLO3/4-UTX complex to non-canonical sites, but not only non-canonical sites. These sites are actually normally bound by the menin MLO complex. So then the next question is, what are the transcriptional consequences of these very drastic molecular switch between the MLO1 and MLO3/4 complexes? So to answer this, we then perform transcriptional profiling of leukemia cells that were treated with the inhibitor. So as I mentioned during the introduction, this complex is canonically associated with coactivation of gene expression. However, when we disrupted using a small molecular inhibitor, we actually observe an up and down regulation of gene expression. And in fact, these changes are actually skewed more towards upregulation, suggesting that perhaps at steady state, this complex is also able to co-repress gene expression. So then to better understand this novel putative function for the MLO1 Menin complex, we then focus on the upregulated genes. And what we found is by integrating both the epigenomics and transcriptomics data sets that we were able to obtain a number of sites that are reciprocally bound by Menin and UTXA promoters from the chip sequencing datasets here in the first gray column. Then the genes that are induced by the Menin ML inhibitor by RNA sequencing, and then the overlap of these two datasets in red. And what you can see here is that the majority of the significantly upregulated genes by the Menin inhibitor are actually reciprocally bound by Menin and UTX, suggesting that actually their expression might be co-regulated by these two chromatin factors. And to the best of our knowledge, these genes represent a previously unknown catalog of many MLO target genes in leukemia cells that are actually repressed at steady state. And these genes are sensitive to the action of the Menin MLO inhibitor, which results in the redistribution of the MLO34 complex to their promoters and subsequent activation of the expression. And lastly, in an effort to prove the human relevance of this new or novel molecular mechanism, we teamed up with the Armstrong Lab and the Dana-Farber to perform in vivo experiments using PDX models of MLO-VRH leukemia. And very quickly, in this experiment, we transplanted mice with an MLO-AF9, MLO3 wild-type PDX, treated these mice with the Menin MLO inhibitor, and then assessed the tumor burden of these mice over time. And what we observed is that these inhibitors are actually quite potent and eradicated these leukemia cells in vivo. However, when we engraft mice with a human PDX model that is actually, in this case, MLO3 mutant, these mice completely failed to respond to the action of these inhibitors, suggesting that really we were able to identify the mechanism of resistance in mouse models of leukemia that were readily validated in these preclinical models. And in closing, the dogma in the MLO fusion leukemia field is that cells are actually primarily sustained by these aberrant transcriptional programs these chimeric proteins are able to regulate, and that when this circuit is disrupted, the cells differentiate and ultimately die. Our work actually shows is that at steady state, a fraction of the Menin wild-type MLO1 complex actually coordinately represses the expression of a tumor suppressive network in leukemias by direct binding to their promoters. And disruption of this complex using genetic or pharmacological approaches actually then triggers a molecular switch between these two complexes that then results in activation of these genes, ultimately killing these cells. And in summary, our work really provides the first evidence of a functional interaction between these mammalian histomatic transferase complexes and supports the notion that these complexes are actually very dynamic and plastic in the functions that they can elicit in cells. And lastly, these are the two major clinical implications of this work. And just really very briefly, I just want to get back to this idea that in order to get a complete and holistic view of chromatin regulation, both in normal development and cancer, we need to understand how these chromatin adaptor proteins are able to help then decode the chromatin landscapes of cells and then regulate transcription in different contexts. And as I move on in my career, I just want to share with you the three main directions that my group will be initially working, trying to understand what are the targeting mechanisms that these adaptor proteins utilize to target the places in the genome that they need to regulate, also how they're able to nucleate specific complexes in very specific settings, and lastly, how cancer-associated mutations then affect these functions. And last but not least, I want to thank everyone who contributed to this work, and inside the Allis Lab, my collaborators outside, funding sources, and you for your attention. Thank you so much.
Thank you, Yadira. That was wonderful. We already have a couple of questions in the Q&A. So I can start with those while people raise their hand if you want to, or type in more questions. So Ben Weekly asks, do Menin and UTX interact physically via co-IP or IP-MAS spec? I guess he's wondering what your hypothesis is for how Menin restricts UTX binding at these sites.
So we think it's really a combination of things. So we think there's actually a repressor within this complex, within the Menin MLO complex, that is keeping that co-activating activity of that complex kind of in check. And we're currently doing those experiments to see what we have. Some candidates in this case is actually HDAC1 and DNMT1, the two that we're testing. So we think association with a repressor is what is keeping those target sites off. Kind of going back to the original question, whether Menin and UTX interact or are part of the same multi-protein complex. So we've done a lot of experiments trying to see whether that's true, and we cannot detect any association between these two. And we think it's truly a displacement mechanism that is at play here. So you have the canonical complex bound at these sites. When you disrupt that binding, then the other complex comes in. So it's really a yin-yang situation that we think it's at play. So we're trying to really dissect what is keeping things in check at steady state. So we think transcription factors is one thing and a repressor is another thing. So those are being tested right now. Great question.
Okay. The second question is from Laila Hedaya. How would you explain the high fold change in UTX occupancy as compared to the Menin occupancy after Menin inhibition?
So there's a couple things. So the levels of UTX, or the protein levels of UTX in both conditions actually don't change at all. So they're very similar. So at steady state and in the Menin inhibitor treated. So we think it's really just redistribution of chromatin unbound to chromatin bound UTX. That it's this massive enrichment of promoters that we're seeing. So we're doing experiments trying to see whether potentially there's some ideas that UTX can be localized to the nucleolus, and then certain signals are able to, translocate it out of the nucleolus and then have its functions or canonical functions in the nucleus. So that's one hypothesis we have, and we're currently testing it. But there are potentially other mechanisms at play as well that we were considering.
All right. And then Pradeepa Madhurapura says, cool work, and then asks, does Menin inhibition lead to increased H3K4 monomethylation or trimethylation at promoters due to the UTX MLL3 binding?
Yes, so that's the data I didn't have time to show you. So we also checked for the methyltransferases in each of these two complexes, MLO1 and MLO3 and 4. And we see decrease of MLO1 at these promoters, decrease of K4 trimethyl, increase of K4 monomethyl, similar to the question that you're asking, and also increase occupancy of the methyltransferases as well. So we think at least the core complex is being targeted to these sites in the context of the inhibitor, yes.
All right, I think we'll ask one last question before we move on. It's from Brian Lau. Sorry if I butchered your last name. Again, he says, great talk. And he asks, along the lines of dissecting scaffolding functions, is the catalytic activity of UTX necessary for activation of Menin target genes?
Yes, it's not. So that's something that we have tested. So we think in this setting is really serving as a scaffold for the MLO3/4 complex. And there's some beautiful work from several groups, Bob Rader and others, that have shown that it's actually the ability of UTX to bind to chromatin directly that it's required for its functions in enhancer regulation. It's not really the ability of these demethylase to remove methyl groups from K27. So in our setting, we also think that it's catalytic independent function as well.
Cool. Well, thank you very much. That was wonderful.